Um, we are going to dive right into this topic right here. Um, if I can get the click. Yeah. So the point of this class is try to start demystifying this idea of plant nutrition. I found that when I started gardening, I read a bit about how to provide nutrition for plants and what we need in the soil for them to grow healthy. But I originally found that the descriptions of why plants needed these nutritions, it kind of left me in the dark, and I was frustrated until I came across this book called Teeming with Nutrients by Jeff Lowenfels. And this book, this presentation is really homage to this book. It really kind of opened my eyes and made it really clear in an understandable way. It's not written for like your expert majors in botany. It's written for more common gardener folk like us. So I recommend, if you like this little presentation, I recommend checking this book out of the library or picking it up. It's really good. And so we're going to explore the idea of what are nutrients actually, how are they used within the plant, and so why are they really that important? And once we get that, then we can figure out how we can help plants get what they need so that they can do what they do and grow and be delicious and nutritious for ourselves. So that's the idea of this class. And so if we were to look at what a plant is and just like weigh it up in its total mass and break it down by its parts, you can see in this pie chart that most of it, 90% is carbon and oxygen. So 9/10 of what makes up a plant physically is actually being breathed in through those stomata from the atmosphere. And then 6% of it is hydrogen, so that's coming in through the root system. And so yeah, so 96% of what makes up a plant is not typically what we think of as being nutrients, but they are, as we'll see, definitely an important part of the plants. They just tend not to be the limiting factor in plant growth. That last 4%, 3% of which is made up of nitrogen, are what we typically consider the nutrients that we need to get to the plant. I mean, hydrogen can be obviously important if you live in a desert or your watering scheme isn't well done. But yeah, so like only that last 4% of what makes up plants are generally considered by gardeners the plant nutrients that we have to worry about. And a little bit of terminology here. We call the NPK is what in common parlance of gardening stands for nitrogen, phosphorus, and potassium. And they are generally considered the macronutrients. And it refers to the nutrients that the plant uses the most of. And so we have to supply those NPK, the nitrogen, phosphorus, and potassium, at much higher levels than we do the micronutrients. There is an argument to be made that calcium, magnesium, and phosphorus belong in this macronutrient category, but you'll generally won't see them show up on the labels of fertilizers. But the plant does use relatively larger amounts of calcium, magnesium, and sulfur. So a little bit of a debate there, but yeah. And then there's also the rest of the plant nutrients known as the micronutrients. And that designation does not mean that they aren't important, because they are still really vitally important. It's just they get used at much lower quantities, um, being taken up through the roots from the soil and incorporated into the mass of the plant. So because they get used at much lower quantities, they tend not to get depleted from the soil the same way as the macronutrients. And so they don't hinder growth, generally speaking, as much as the macronutrients. So in effort to streamline this presentation, I'm going to focus pretty much mostly on the macronutrients, but I don't mean to ignore the micronutrients. They are still really important. And so when we think about the fertility of the soil, there was this gentleman in the 18th century or 1800s called Justus von Liebig. I think I pronounced that right. And he is considered the father of the fertilizer industry. He was really, really big into organic chemistry, and he came up with, not only did he really kind of invent synthetic fertilizers, but he came up with this idea of the law of the minimum. And his point with this is that the overall amount of all the nutrients isn't so much as important as it is the lowest one, the least, the nutrient that is the most scarce in the soil will slow down plant growth and affect. It'll be the limiting factor. It doesn't matter how much of all the other nutrients you have in the soil. If you're lacking in one nutrient, as you can see here, his analogy of the barrel, and S stands for sulfur. So this is just the example. But if that's the lacking nutrient that is scarce in the soil, it doesn't matter how much phosphorus or nitrogen or boron or zinc you have, that lacking nutrient is going to slow down everything. So something to consider there. And then another really important figure in the history of gardening and the history of understanding plant nutrition is this gentleman, Sir Albert Howard. Just really like this picture of him, that steely-eyed look in his eye. Look like he's really dialed in there. But he came up with another rule, which is the law of return. And so what we take out of the soil, we have to put back. And so as plants grow, they're absorbing those nutrients from the soil. And if we're harvesting plants out of the soil, then those absorbed nutrients we're taking out of the soil. And if we don't replace them, then the soil will become increasingly depleted, and our future plantings will become less and less productive. And this is something that you can't cheat. I learned this lesson the third year I was gardening with the school garden programs. The second year, or actually, I guess it was the fourth year. So yeah, in the third year, we built a whole bunch of infrastructure at the middle school. We built a tool shed and a greenhouse, and we ended up going over budget on the greenhouse. So the next year, I tried to be cheap with replacing the nutrients that we had pulled out of the garden from last year when we were harvesting. I did not invest in fertility that year, and it was a very frustrating growing season for me, because we did all the work of preparing the beds and planting everything, but because we didn't replace what we took from the soil from that past year, plant growth was really painfully slow, and it just wasn't worth it. And that was the time when I learned the lesson, like, if you're cheap when it comes to soil fertility, you're going to pay, it's going to be more expensive. You're going to pay for it in your time and your work. So it's really worthwhile knowing how to keep the soil fertile, and that'll make your time more efficient. So this next slide is just as we start to try and wrap our minds around what essential plant nutrients are. I just kind of like to look at this because on the left of the screen is the periodic table, and we see all the atomic elements of physical reality. And a chemist will look at this and just look at elements. And then on the right side of the screen is a list of the essential plant nutrients. And you can see how all the essential plant nutrients are indeed found on the periodic table. But whereas the specialized chemist thinks about them as atomic elements, the gardener or the farmer or the agronomist thinks of the same thing as essential plant nutrients. So we're going to look at how the plant is using these to function and to grow and to be healthy. And so here is a diagram of a generic plant cell. And so we're going to dive in order to understand how the plants use these nutrients. We're going to have to look at the actual structures of the plant cell themselves. And this is a generic representation. And I say that because the actual physical cells within a plant can vary from cell to cell depending on their specialized functions, as we'll see. You can see here we have our cell wall on the, I should get my pointer going. Nah, never mind. I don't know where it is right now. But yeah, we have our cell wall here on the outside, and that's usually made up of your cellulose, and in woody plants, there'll be the lignin material, which we tend to think about as wood. And then we have our cell membrane here, which would be the yellow portion. And we have a whole bunch of organelles within the cell. I'm not going to go over all the organelles because I figured it would be kind of drawing it out. It would be a little bit tiresome to go through all of them. But you can see here the Golgi apparatus. I'm not going to talk about beyond this point. That's basically like the packaging. It's kind of like a mail office. It's responsible for packaging up materials and sending them on their way where they belong. You got your peroxisomes, and so that is like a recycling center. I'm not going to talk about that anymore. We got our endoplasmic reticulum over there. That does a number of different things, So, but they can all change based on that specialization. And then you have your vacuoles, and then this structure down here, I'll talk more about. That's the chloroplast. And so I think it's just really interesting to point out right now, we think of ourselves as animals, as humans, as being really different from plants. But I want to take this opportunity right now to point out that other than the chloroplast, the vacuole, and the cell wall, everything else you see in here is we share in common at a cellular level. Everything other than those three, we have all the same organelles and all the same structures, and our cells work very, very similar. So we tend to think of ourselves as being super different, but more things in common than we have different, surprisingly. And then I just want to point out that, just to give you an idea of scale, a micron is 1/1,000,000th of a meter. And so the average plant cell is about is 1/1,000,000th of a meter, a micron. So that's the size that we're talking about. And then all these organelles in here that do all these specialized tasks for each and every cell within a plant is measured by nanometers. And so a nanometer is a billionth of a meter. So super, super small. And then for one more point of reference, a human hair is 100,000 nanometers thick. So we're just the level of complexity that we're going to talk about here, and the level of microscopic is really kind of mind-blowing, and something that I really appreciate, and I hope you do too. So I want to, talking from the standpoint of that NPK that we're going to see on the labels of every fertilizer. Let's start with nitrogen, because it was after that carbon, oxygen, and hydrogen. Nitrogen is used most in the plant, and it's really, really important for the plant, and I'll explain why. So nitrogen is the key component to make these things we call amino acids. And amino acids are chained together to make proteins. And so proteins are the workers. That's why I got this picture of the workers on the skyscraper back in the day. They pretty much do everything. They build everything at a cellular level. They transport things. They metabolize, and they perform storage tasks. So proteins, basically, there would be no plant growth to speak of if there's not enough nitrogen in order to build these amino acids and then build the proteins. This is a representation of what we think a protein looks like. And so when you see that, that is just long chains of amino acids, and within the amino acids, we have that nitrogen part that's making up the molecule that builds those. And proteins are really interesting and mind-boggling and kind of simple at the same time, based on the way that those amino acids are built together, and these side chains that get put on them. They will bend and fold in really unique ways, and parts of the proteins are hydrophobic, so they don't like water, and other parts of them are hydrophilic, and so they do like being exposed to water. And this relationship between that, just their preferences for exposure to water, is what makes them fold and move, and makes all this work that happens at a cellular level happen. And it's really simple, but it's also extremely complex as well. Proteins, we're starting to kind of understand them as human beings, but we don't really fully understand them. And my point to prove that would be, if we really did understand everything there is to know about proteins, we wouldn't have had this pandemic happen this past year, because a virus is just diffuse DNA surrounded in a protein coat. And so this virus has just absolutely brought the whole global economy to a standstill almost, or at least it did severe damage there. And it's crazy to think about how the way these proteins fold in these unique manners is ultimately what's responsible for such devastation that's gone on over this past year. But I should digress from that. But anyway, so nitrogen, really important, because it's basically the building block for these proteins, and the proteins pretty much build everything. They're responsible for growth. Enzymes are a special kind of protein, which they act as catalysts within the cell, and they speed up a lot of really important chemical reactions in the cell. And so as an example, some of these chemical reactions that you might be familiar with already is photosynthesis and cellular respiration. So if it wasn't for the protein, these specialized proteins that we call enzymes, these really important chemical reactions would take place at such a slow glacial pace. We wouldn't be able to grow. I mean, we wouldn't. Plants wouldn't be able to grow. We wouldn't be able to grow food, and we wouldn't be able to grow ourselves. So, I really appreciate enzymes and proteins and the nitrogen that makes it happen. And then at a cellular level, there are these ribosomes which are in charge of building the proteins. And so you can see here in this little diagram, the amino acids go into the ribosome, and the ribosome is reading the RNA messenger basically script that they get from the nucleus, and that's what tells them what kind of proteins they need to build. And so when you see the ribosome in the diagram of a plant cell or even a human cell, that's what it's doing. That's the factory for building the proteins. So really important. An average cell has about 1,000 ribosomes, but some cells can have multiple thousands, just depending on what their specialized role is. The next really important thing that we need nitrogen within the plant cell for is that they are, you can see if you look in here, they are part of the structure of all of these nucleotides that make up the DNA, which is basically the script that the ribosomes have to read in order to make the proteins which do everything. So we can think of the nucleus as sort of like the master architect that makes these blueprints that are read by the ribosomes, and the ribosomes make the proteins, and the proteins go and do the work. Or you can think of the nucleus as somewhat of a dictator, because it's basically telling everybody what to do. But yeah, so I'm sure everybody out there is familiar with how important DNA is to the functioning of the healthy functioning of a cell, and then how that can kind of be extrapolated out to the healthy functioning to an entire organism. So if we don't have those nitrogens, then we can't build the DNA, and so nothing really happens. And then it's also an important part of that messenger RNA that's getting sent to the ribosomes. And so those are obviously two really important things. The next really important things at a cellular level, especially for plants, I mean, it kind of makes a plant, is the chloroplasts. And the chloroplasts are the site of photosynthesis, and what makes photosynthesis happen within those chloroplasts is the chlorophyll molecule. And so this is a diagram at a molecular level, what a chlorophyll molecule looks like. And we can see as we get right to the middle of it, we can see those 4 nitrogen atoms holding the magnesium atom right in the middle. And so again, it's pretty obvious if you look at it this way. If you don't have enough nitrogen, then you can't make the chlorophyll, which makes the sugar, which is like that. It's that autotrophic. It's what makes plants plants. So another just pretty clear example of why we need to make sure that nitrogen is not in a short supply here for our plants. Okay, so I'm not going to beat a dead horse. I'm sure everybody out there can see it. Nitrogen is really, really important. But my question for you, if anybody wants to ask, is where is the nitrogen? Where do we get it from? Yeah, Does anybody want to take a guess? Where is nitrogen?
Feel free to raise your hands or type it in the chat.
Yeah, yeah. I'm going to take a nice drink of water.
Deb, what's the answer?
Well, I know you can get it into the soil depending on what you plant there. Like you can plant certain plants that help to fix the nitrogen in the soil.
Yep, yep.
So I think it's probably already there, but maybe not in some way that can be used. I don't know. That's my guess.
Yeah, okay. Anybody else want to take a guess? All right. It's a cliffhanger. Let's
You got some in the chat here. Hold
okay.
on.
What we got?
Brooke is saying the air. Maybe.
Yeah. Yeah.
Okay.
Good job, Brooke. Yeah. So nature has a very twisted sense of humor in this case, because roughly 78% of the air we breathe is actually nitrogen. It's in this form of atmospheric nitrogen. So it's so ironic that the thing that we need to grow, not just plants, but ourselves, everything is so in desperate need of nitrogen. We're just swimming in it, but it's in this form. This is the molecular kind of model showing that it's in this form called N2, and basically we have two nitrogen atoms here, and a crash course on chemistry. If an atom can be happy, then it's happy to the extent that it can have 8 electrons in its outside orbitals. And so that's what makes a lot of our molecules. That's what turns a lot of the molecules that we know, whether it's oxygen that we breathe, O2, or it's H2O, or it is carbon dioxide. It's what creates this attraction between atoms and gets to us to the point where we go into molecules. And so you can see here that in order to get to 8 electrons on both of their outer orbitals, they're sharing the electrons there to make their 8. And the fact that this is a triple bond makes them really happy in a sense, and makes it very stable. It makes it really hard to break. So it's all around us, but it's basically inaccessible. And for the vast majority of natural history, the nitrogen that plants needed in order to create these proteins so that they could grow, it came from microorganisms and from lightning. So because that bond is so strong, consider the amount of energy that happens in a lightning strike. That's how much it takes to fix nitrogen from this atmospheric form into a soluble form that a plant could use. And then what an animal, well, like once the plant gets it, then the animal comes in and gets the nitrogen and proteins that they need. So some of it has been converted from this almost impossible to grab form in the atmospheric form by lightning, but microorganisms, like, this is kind of blows my mind. It never stops to fascinate me that if you consider how much energy is in a lightning strike, a microbe, this is a picture of Rhizobia. So it's a nitrogen fixing, that's a whole colony of bacteria that forms root nodules on legumes. There are other microorganisms that can do this as well, but the most well-known in gardening, and just to keep it simple for today, we'll just talk about Rhizobia right here. They just have this really specialized enzyme that is able to break this triple bond and turn it into a form that they can use. And so that's all leguminous plants have this ability, and that's what makes that family of plants so special. And so for most of history, this is how it's happened. It is leguminous plants, and there's also Frankia, which associates with alders and sea buckthorn. But yeah, so that is how it was for millions and millions of years. That's how nitrogen became available to the living world. And then humans figured it out. So if you think of like food producers in the past, in traditional ways, they would use like animal manures as a viable source for supplying nitrogen. There were, I can't remember if it's the 16th century or the 17th century, Dutch farmers finally put 2 and 2 together and started to grow leguminous crops on purpose, because they realized that they were able to fix nitrogen and it made their soils more rich. But for the vast majority of human history, we just kind of relied on recycling animal manures in order to supply that nitrogen to the plants and grow them. But as population levels increased dramatically with the industrial age progressing, there became really large concerns about diminishing supply for soluble nitrogen. And people started looking out for more and more sources. And this picture up here, this is a picture of a guano mine in Chile and Peru. They just had really, really perfect conditions. If anybody is familiar with the Humboldt current that goes down the Pacific coast of South America, it makes a very dry climate. So, and then on top of that really dry climate, not very much rain whatsoever, the currents of the ocean were very favorable for fish life. And so that attracted birds, and there weren't any predators for the birds and the bats. And so they proliferated on these islands off the coast of Chile and Peru. And these are bird shit and bat shit mines. And so they started like some of these mines were apparently like 200 feet thick. And I didn't have time to put all the stats about it, but I think I remember reading that around this time when they started mining these Chilean guano mines, like something around 13 million tons were shipped out of here. And so that was the source for a lot of production as populations started to grow. And but people were really, really concerned that they were going to exhaust these sources for nitrogen. And there was actually there was an escalation of military might around these mines. And so look up the guano wars. There were actually countries willing to go to war with each other over bird shit and bat shit. So history can be funny sometimes. They weren't just interested in the fertilizer aspect of these nitrates, these sources of nitrates. It's also a key component to building bombs. And so there's a little bit of a weird history here between nitrogen fertilizers and both guano, and then the invention of synthetic fertilizer. There is I don't know if I can call him a gentleman. There's this genius, a mad genius named Fritz Haber, who he was interested. He was a German nationalist around the World War I era, and he was just absolutely blow your socks off smart in organic chemistry. And so he wanted to solve this problem for the nation of Germany. And so he came up with this process, this way of synthesizing nitrogen fertilizers from the air. And then it was industrialized by this guy, Carl Bosch. And so this is a little representation of what the Haber-Bosch process looks like. It is quite energy intensive. You need around 500 degrees Celsius temperatures. You need a certain amount of atmospheric pressure. Like you need to increase the pressure within these tanks. It needs to be, there's a catalyst of uranium and I forget the other one off the top of my head, but he found out a way of synthesizing finally, of creating a man-made way of industrially producing synthetic fertilizers. And so this changed the game. They won a Nobel Peace Prize for it, which is ironic because yes, he invented synthetic nitrogen fertilizers, but at the same time, he did that in the interest of trying to find a way to make more bombs to kill people. I'd like to tell you more about Fritz Haber, but there's a YouTube channel called SciShow that did a profile on Fritz Haber and his mad genius, like absolutely evil mad genius, that I really recommend looking up. SciShow, Fritz Haber, look it up. It's really cool. And so the invention of nitrogen fertilizer is very, it's both a blessing and a curse at the same time. Like I said, there were real concerns about mass starvation because they thought that our food production systems would be stifled by a lack of nitrogen, and they thought just millions of people would die. So it saved I think the figure I remember off the top of the head is that 1 in 5 of us wouldn't be here right now if it wasn't for that. So on the one side, that's good, but on the other side, the cost of these nitrogen fertilizers is something that we need to think more deeply about. About 5% of total natural gas use every year goes towards the production of these fertilizers. And so, yeah, they're quite energy intensive, but when we rely too much on them, we rely on a fossil fuel that is contributing to climate change. It is a finite resource. So as we start to use up this finite resource, it's going to cost more and more to make it. And so if we continue to rely on it moving forward without thinking about alternative ways, then you will see there'll be a correlation in the cost of food, because if the fertilizer costs more, then our prices are going to go up, and that's not a good thing for food security. There's also an issue with eutrophication. So when nitrogen fertilizers are applied to the field, they are in a soluble form. So if a farmer irrigates their land too much after applying that, or we get a lot of rainfall, then that nitrogen fertilizer and the phosphorus fertilizer, which can be synthetic as well, gets washed through the soil, and it makes its way into natural marine environments. And in nature, you'll never find a concentration of nitrogen and phosphorus at that level. So they contribute to these huge blooms of algae, particularly in the Gulf of Mexico at the mouth of the Mississippi River. Every year, you have these massive, massive, like 20,000 square kilometer blooms of algae, and that chokes out the life. First of all, the light can't get down into those marine ways, so it kills off a whole bunch of life. And then once the bloom has, like, once they've used up all the available energy there, then they die off. And as they're dying off in an anaerobic environment, it basically makes it takes all the oxygen out of those marine ways, and it kills all the marine life. So on the one hand, I'm thankful for nitrogen fertilizers, because 1 in 5 of us being here, like, I'm a big fan of everybody being here. But then on the other side, it has come at a cost to the environment. And I think I'm not ever going to sit here and tell people what kind of fertilizers to buy, because that's not my place. But I offer this information for people to make more educated decisions for themselves. So that's a little spiel about nitrogen fertilizer. Any questions about that?
I think you're good to go.
Okay. So yeah, so here's if we're gonna try to use other sources to fertilize our garden, I'm gonna talk more tomorrow, or sorry, not tomorrow, but next class. I'm gonna talk more about how we can use composting methods, and how we can work with those leguminous plants in order to provide for the nitrogen for our garden. But here's just a little list here of options, more natural options that you might look into. So we have Alfalfa Meal, which is there because it is, so this is a picture of Alfalfa Meal. Alfalfa, as we talked about last class, is a legume. So it's basically farmers are growing alfalfa and then just chopping it up into really small forms and selling it as a meal that you would use as an amendment to add nitrogen to your soil. Bat guano, I've never actually seen this around these parts, so good luck finding that. But it is considered a natural source. I don't know how people would feel about using that after the whole COVID year we've had too. But anyways, blood meal is another really good option. It's soluble. So when I was talking last class about producing early in the season when it's cooler, using a soluble natural source for nitrogen is going to promote plant growth, rather than if we use compost in the cool part of the season. Compost really relies on the soil food web cycling nutrients. And because it's cold, the soil food web kind of works at a slower pace. So if you're looking for early season production during the cool shoulder seasons, either early or late, then I would recommend blood meal for that. On the negative side for blood meal, it does come from the basically like industrialized, it comes from basically like the slaughterhouses that do not treat the animals with much dignity whatsoever, to speak of whatsoever. So that is like the negative side of it. I don't really like supporting this industrialized meat farming model, but it is a more locally sourced, so none of this is to say what's good or bad. It is what it is. And we just talk about it so that we can try to find more sustainable, more humane ways of growing the food that we like. So here are a couple other examples that you might be able to find. And then I have fish emulsion. There's also fish hydrosylate. I don't really recommend, due to our wildlife, due to the bears that we have in the area, this might be considered an intense attractant. It is an option, but given our proximity to the bears and our not so fondness of having wildlife human conflicts, then not such a good idea. This is the surprising one. Human hair is like, that's one of the highest ratings for available nitrogen that I can found, but obviously it's kind of weird putting our own hair, mixing our own hair into the soil to grow our vegetables. But again, it is what it is. And then we see human urine is actually quite rich in nitrogen, and it's got a little bit of phosphorus and potassium. It's not recommended to urinate around your food crops, though, due to if antibiotics and mercury and heavy metals and stuff like that, which begs the question, why do we have that in our own bodies to begin with? But again, it is what it is. And then soybean meal, nice source, more natural. Soybeans are in the legume family, but I would say as the detractant, it's grown in huge industrial size monocultures, which basically eliminate every other plant within how many thousands of square acres. And so that's not necessarily environmentally regenerative either, but those are the options that we have. You can leave comments in the comments section to let us know what you think about those. And again, I did mention that I'll talk more about compost and legumes in the next class. So let's move on to the next one in the NPK triad. Let's talk about how phosphorus is used at a cellular level. Every cell membrane, so the outside cell wall is like this cellulose, it could be lignin, it's like this more tough structure that kind of gives them the ability to stand up straight and reach for the light, but still have enough flexibility to bend in the wind. And then within it, we have our cellular membrane, which is kind of like the gatekeeper. If there's anybody younger in the crowd, this is a Ghostbusters reference from the 80s. But we kind of think about the cell membrane as the gatekeeper. They decide what comes in and out of the cell. So obviously we're made up of a lot of cells, as plants are, and so there's going to be a lot of these cell membranes. And you can see here again in our molecule, kind of look at it, at their heads there's always going to be a phosphorus atom in there. So if you think about all these heads that make up all the cell membranes, that's a lot of phosphorus that a plant requires. And I also wanted to use the nerdy scientific name for it, plasma lemma, strictly because it kind of points out that every cell membrane is made up of plasma, which isn't a solid, liquid, or gas. It's all three at the same time, which is kind of cool, because other things that in the natural world that are plasmas, that are all three phases of matter at the same time, are lightning and the northern lights, like aurora borealis. So I just like to think about how each one of my cells has a shared characteristic of the northern lights. And so as you can see here in this diagram, we can see that the head is hydrophilic. It will face outwards towards the water, whereas the tails of it, you can see it in here, are hydrophobic, and they'll always point in away from it. And so when we think about our cell membranes and plant cell membranes, is think about the way oil kind of coagulates in water. It's the same kind of pattern, if that's helps you at all. I hope it does. And then since we're here, since we're looking at this diagram, the plasma lemma, or the cell membrane, is studded continuously with these transport proteins. So we see nitrogen showing up again, how important it is. Water is able to pass through via osmosis, but for bigger molecules or materials that need to get in and out of the cell, these proteins play a really important role.
Christian, before you move on, we got a question and a comment in the chat. And I think kind of going back to the previous slide about the fertilizers, Brooke's wondering, have you heard of, I'm not sure if I'm saying it right, okara? It's a byproduct of the tofu industry.
I have not. It's a byproduct? Okay.
Brooke,
Well,
do you want to comment?
Yeah. So I live on Vancouver Island, and a lot of the farmers use it here.
Okay.
It's just really high in nitrogen.
Yeah.
But it is definitely a wildlife attractant as well.
Okay.
So it's tricky. Yeah.
Yeah.
Yeah. And that's interesting about fish emulsion, that you can't use it as well in Canmore.
Yeah, that's the. I've never used it, so I mean that's just what I've read. So I'm sharing that, and it makes sense. But yeah, it is okara.
Yeah, okara.
Okara.
Yeah.
Okay. And it's a byproduct.
There's a. Yeah. Yeah, the farmers get it in like big barrels,
Okay.
but it's really heavy. Yeah.
Yeah. Okay.
Yeah, but it's
Easy
really
to
good
buy.
for brassica.
Yeah. Okay.
And it's free. Yeah.
Cool. Well, yeah. Like, so if it's a byproduct, I mean, it kind of goes with blood meal. I mean, it's different, obviously, but like, if it's a byproduct, then it's nice. We don't want to waste anything, you know. So that's a plus, in my opinion. And so I obviously, anybody. Oh, can you turn your microphone off? Because I'm getting. Yeah, thank you. Yeah, so like, yeah, anytime we can find a byproduct or a potential waste and put it to a new use to grow new plants, I think that's a plus. And obviously what I said about the industrialized factory farming model, we don't really want to support that, but it is a byproduct because it's a waste product. And so why would we let it go to waste, you know, when we can recycle it? So yeah, there is kind of lots of gray areas there, and I think that's interesting. Thanks, Brooke.
Yeah, thanks for sharing that. And then Katerina just had an interesting comment that she has used urine diluted with water for her peonies many years ago, and the blooms were amazing, and now she knows why.
Yeah. Yeah. Okay, so we'll move on to phosphorus and its importance for cellular respiration. So the organelle within the cell called the mitochondria is something that you could think of as akin to the powerhouse of the cell. And so this might seem counterintuitive because we tend to think about plants, where plants get their energy from, as being through photosynthesis and the chloroplasts and the sugar that they create. But that isn't the currency that the cell can use in order to carry out its tasks. The sugars created from photosynthesis are what build, this is called the adenosine triphosphate. And so this is the molecule that is responsible for creating the actual energy that the cell can use. And so the sugars created in photosynthesis, they add this third phosphate group onto this molecule, which beforehand it would be adenosine diphosphate. And so when the sugars are used to add this on, then it can break apart again. And when it breaks apart again, that's what creates that cellular respiration that not just plants use, but every cell has this cellular respiration. And so that is really, really important to kind of understand the distinction between the way that sugars make their own energy, but they convert that sugary energy into building adenosine triphosphate just so they can break those molecules again and get the real energy that they need. Seems a little bit inefficient, but it's pretty awesome. And so the point here is that 1, 2, 3 atoms of phosphorus for every single one of these adenosine triphosphate molecules. We can see pretty clearly why the plant needs phosphorus in a larger quantity than the micronutrients. And then we can also see there's some nitrogen over on this side chain here. So again, nitrogen gets used a lot. What else do I got over here? Yeah, so there can be, each cell needs a lot of this energy in order to do what it needs to do. And so there are hundreds and even tens of thousands of mitochondria in each cell. And that's where this cellular respiration, that's where the forming and the breaking of the adenosine triphosphate is going on in order to make that energy. And one of the ways to spot a phosphorus deficiency in your soil is that the older leaves on your plant, if you have a deficiency, they'll start to get this bluish kind of tinge to them. And that's because they're getting buildup of sugars, but they don't have enough phosphorus to translate that into the energy that they need as a plant. So if you ever see a bluish tinge to your older leaves, then it's a pretty good sign that you should look to add more phosphorus to your soil. And then we got to come back here. Well, actually, you know what? I forgot to say this. The other word for the cell membrane there is it's phospholipid bilayer. So just that name right there should indicate how much phosphorus we need. And then again, we're coming back to this DNA molecule, and you can see how the nucleotide backbone is at the center of it. We have these phosphate groups that are holding these ones in place. So lots of phosphorus we're using. And then we'll go talk a little bit about our natural sources for phosphorus. So animal bone meal is a place to get it, and so you can buy bone meal in garden centers. You can buy little packages, but you can also make it yourself. There is all sorts of recipes out there on the internet if you want to look up. I don't have time to explain that whole process, but you can save up the bones. You can make a bone broth first in order to maximize those bones, make a nice flavorful, nutritious broth, and then you can save them up, and you can process them in order to make your own bone meal. Otherwise, when you buy the bone meal, again, we're kind of supporting that industrialized factory animal farming model, which isn't the best. But again, it would be considered a waste product. So pluses and minuses. In my experience, I've used bone meal And I guess at my scale, I find that buying like these little cartons of bone meal or blood meal, like it's just way too expensive. So I started bugging a couple of years ago. I bugged this company called Terralink for several months in order to buy big 20-kilogram bags that were way better value. And so if anybody's out there is interested, I had to bug the people at the head office for a couple months, but they finally sent me out a whole bunch of bags. And when I got to the warehouse, the guy said that if we call the warehouse directly, then he would sell to us. So I don't really see really good value in these little boxes that you can get, even though you should support local businesses. But it's marked up way too much. So I would suggest if people are interested in bone meal, then going in together in order to buy a couple of these bags and then splitting it up is the option that you want to look at. And there's a kind of bat guano that I know I said earlier, and I had a different NPK value there. There is a type of bat guano that is higher in your phosphorus than it is nitrogen. And then there's like rock dust, like rock phosphate colloidal dust that you can buy. Not a high percentage, but it'll last a lot longer in the soil. And then crab shell meal. We don't have a lot of crabs here, so and I've never actually seen that in the store, but it's another option. There is a synthetic option. So there are rocks called rock phosphate that they are basically like dissolved in acids in order to make those phosphates more available. And so that's the synthetic option. If you're interested in going that route, then that's your call. But the thing about the phosphorus in that medium is that it gets binds up in the soil readily, and so you can spend a whole bunch of money on this rock phosphate or the superphosphate synthetic version, but it's only available to the plants for a very short period of time before it binds up and it's not available to the plant again. So I would urge caution in using it, because you can spend a lot of money and not get your money's worth. And then there's also this concept, it is debate, it's up for debate, is this idea of peak phosphorus. The Global Phosphorus Research Initiative estimates that the supplies of this phosphate in this form will be inefficient for crops within 30 or 40 years, and there could be another kind of intense military buildup, because this is such an important plant nutrient for growing food crops for people's that yeah, there is a chance that our supplies of phosphate in this form that we're a little bit way too over reliant on at this point, we could be coming to the end of the era where that it is available. But on the flip side of this, and I'll talk more about this not tomorrow, why do I keep saying that? Next class, that mycorrhizal fungi that I talked about in the first class, the fungi that can allow trees to communicate with each other and share with each other, and around 95% of all plants make this this association with the fungi. They're really good at going out and getting phosphorus from the soil. And the plant will really happily make those trades. It loves to just photosynthesize and trade those sugars that it can make for that really valuable phosphorus. So thinking about ways of making sure we're not disturbing that mycorrhizal association is important. And there's also these phosphorus solubilizing bacteria that can do it too. So when we talk about how plants aren't good at getting the phosphorus out of the soil, the mineral soil, they do have partners that are really, really good at it. And so we should think more about how we can make favorable conditions for them. And then the last one in our NPK triad is our potassium. And potassium is kind of interesting because if you look at cellular biology, it's not used as like a constituent for any of the organs or organelles. It's not a part of any of the structures, but it does play a really, really important role in regulating chemicals. They're one of those protein transport proteins in the cell membrane is called sodium potassium pump, and so it's not part of the physical structure, but it plays a role in the opening and closing of that pump. So very, very important in that point of view. And then there are many enzyme reactions that the presence of protein is really, really important. And so if the potassium isn't present in enough forms, then those those chemical reactions that are depending on enzymes, they'll slow down significantly and affect the growth of your plants. And lastly, it's really important for when we talk about those strategies for plants sending extra sugars down into the root zone, then potassium plays an important role in that the transformation of the simple sugars into starch and moving the sugars around. And so some sources for potassium, green sand is a kind of mineral, kind of rock dust that you can use in order to add potassium to your soil. And also wood ashes. If you make sure that you're not burning any treated wood or any woods with glues in it, then wood ashes is a great source for potassium. But the one thing I would add to using that you should be aware of, if you use wood ashes, then it can push your soil pH in an alkaline direction. And so if your soil is already alkaline and you don't want to make it more alkaline, then that's not what you really want to use, unless you use it in association with something that will kind of offset that shift in pH. I'll talk more about that later. And then there's potash, sulfate of potash. If anybody in town has ever been stuck with a train crossing by Railway Avenue, then you'll know that you see these rail cars going by from the east to the west. And there's a lot of potash that we, as a country, it's a natural resource that Canada is very plentiful in Saskatchewan, and so we will ship a lot of that out to mostly China. So you'll just see a lot of potassium going out that way. Sometimes called Sopomag because it has sulfur and magnesium in it as well. And so potassium is mobile in a plant, so plants can move nutrients around, and so you can see a deficiency in potassium in the older leaves, and you'll see little dead spots. So watch out for that. That should be a pretty good sign that you're lacking in potassium. And also if they start to show signs of wilting, then potassium plays an important role in the regulating of the stomata in the leaves. If there's not enough potassium, then that stomata doesn't open and close properly, and so you'll start to see wilting of plants. And I don't want to, I think everybody's kind of getting the gist of this. I'll copy and paste these last three. These are borderline macronutrients that you need to be worried about. But I don't want to run short on time for the rest of this. So I'll copy and paste this and we'll send it out as notes and you can review that. And then so we talk about the pH of the soil, which stands for potential hydrogen, because pH is a measure of the concentration of hydrogen ions within a solution. If there is a high concentration of hydrogen ions, then that makes the solution acidic. And on the flip side, if there's a lower concentration of these hydrogen ions within a solution, that makes it alkaline. And please, nobody ask me how that works, but because I'm still trying to wrap my mind around it. But it's just the physical reality. I have not come across a good reason to understand how that works, but that's just the way it is. But what you really have to know about pH in soil is that it affects the plant's ability to uptake nutrients. If you have a really, and this is why I like this diagram, if you have a really acidic soil, if you're in this range, then all these macronutrients are harder for the plant to absorb. And same with the other side. Most of the garden plants that we would grow in an annual garden setting prefer just like the slightly acidic to the neutral, as you can see. And so that's why we want to be conscious of where the pH levels are in the soil, because if they're off, then they're not, your nutrients can be there, but they're not, the plants aren't able to access them as easily as if it's in the neutral range. And so if your soil is too acidic, then you can add the agricultural lime products, which will push it to a more alkaline area range. And then also I said the wood ash. And then on the other side, if your soil, if you test your soil and you find that it's on the alkaline side, then you can push it back to the more neutral, slightly acidic range by adding peat moss, or there's like aluminum sulfate that conventional growers grow. And because conventional growers generally use synthetic fertilizers, they don't have a problem with alkalinity. They generally have a problem with acidity. So synthetic fertilizers tend to make your soil acidic. And that's why agricultural lime products are used so much and in so much of a demand. But I will talk in next class about how another way of adjusting your pH. But I hope you don't mind me leaving it as a cliffhanger in the meantime.
Christian, Jack was wondering, and I don't know if you were going to address this in the next class, but he's wondering, how do you test pH in the soil?
Yeah. Next slide, Jack. Yeah, so you can find home testing kits. The ones I've always bought are called Rappi tests. The last time I checked, you can test your pH, your nitrogen, your phosphorus, and your potassium. You get 10 of these capsules for each of them. So they say 40 tests, but it's really 10 tests for each of those variables. So yeah, like $20, $25, I think is what they would come in at, which is pretty decent. And you can get a fairly good idea of what's going on for those macronutrients and your pH, just doing those. Pretty simple to do, and does not take a lot of time either. You might, if I don't know if any like balcony gardeners would find any use in this, but it might be a good idea to get a lab testing of your soil, yeah, depending on your scale and your goals and what you're seeing going on in your garden. But if you do choose a lab test, then I recommend this lab here in Coaldale, Alberta, called Down to Earth Labs. They'll do a really basic NPK test and pH for around $40. And then if you want to get more of your micronutrients tested, then it can range up to $80, maybe $100. I wouldn't do that every year, maybe just a couple years. Yeah, and again, depending on your goals and the scope of what you want to grow. That would be my recommendation. And then I recommend, or I've read, and it makes sense to me, that the best time to test your soil is in the fall, after everything's grown. And so that gives you enough time to kind of consider the results of those tests and consider what the best option is for those amendments in the spring, moving forward. So then you can, yeah, you can add those amendments, and you can, yeah. I also recommend tracking it, keeping like a little garden journal for a lot of other things, but among of all the things you can be putting in those garden journals for observation, is tracking how the nutrients, how their availability and their how much you have in there. You can see the trends in them. Sometimes you might over fertilize too, and so you can kind of get an idea over the years about how much you should be adding, because yeah, you can over fertilize. That's the thing with the other thing about synthetic fertilizers, is they're so potent that if the plant's taking up has way more nitrogen available and phosphorus than it would normally ever get in a natural setting, then the concentrations of nitrogen are higher in the leaves, and insect pests can actually detect that concentration, and you'll have more pest pressures if you over fertilize. So something to consider there. It's not just slop on as much as you want. And yeah, so that concludes my talk about kind of how nutrients are, at least the macronutrients, are found in the cellular biology of plants. Let me know if you found that was helpful. I know when I read this book, it kind of changed the way I looked at plants, and I found it really helpful. But yeah, if anybody thought it was just kind of superfluous, I'd like to know that, or if you have any questions. I'm going to spend the rest of this class, if there aren't any questions, talking about
question. What's a sign that you've over fertilized? What will happen to your plants' leaves?
Yeah, I mean that increased pest pressure, I would say, is that sign. I guess again, you're not going to run into this with a natural fertilizer, something that's not, you won't, because you saw all those NPK ranges. The highest was human hair, which was 18, and that doesn't become immediately soluble the same way as a synthetic nitrogen fertilizer. They range up to like 45 and like 46. They're really, really intense. So when that's over applied, you get leaf tip burn. I guess that would be the thing to look out for if you're using. And I guess also human urine is quite intense. If you were to fertilize with even diluted urine that's too concentrated with the same plant, you'd start to see some burning. It would kind of look crispy at the end. So.
Okay, yeah, thanks. That makes sense. And kind of just what you were talking about, Ekaterina was wondering if it is possible to over fertilize with bone meal or blood meal. But sounds like it's a little more difficult.
No, yeah, not really. Not from the standpoint that it would harm the plant beyond, like, there could be increased pest pressures. But it could be a waste of your money because they are more soluble. And so that means that if you apply it to your soil and you water them in, if you put too much, then it could get washed through the soil before the plant roots have an opportunity to take them up. You'll see on any packages for the blood or bone meal, you'll see they have application rate recommendations. And so you would just stick to those. Okay, yeah, so I think I got enough time to talk a little bit about plant tissues. This is going to be the shorter part of it. But so we just talked about plant cells, but plant tissues are groups of specialized plant cells that are organized into kind of these groups of organs that carry out specialized tasks. And so I'll just talk more briefly about this. Like, we've talked about plant structures and functions and cells now, and now this is just like the last kind of layer of plant physiology that we really need to talk about before we can get into some of the how-tos. And I think it's really illuminating. And I hope you will too. So dermal tissue, we can categorize groups of cells into dermal tissues. And so this is the sort of epidermis of the plant. It's the outside layer of the plant. On the top surface of leaves, they tend to be, but don't necessarily have to be, it's more waxy, kind of a cuticle. It tends to be like this really clear layer of cells that allows light through, but it protects against water loss, and it protects the plant like from injury a little bit. So on the surface of leaves, we would call the dermal tissue the epidermis. But then another specialized cell that gets categorized as a dermal tissue is the trichomes. So if you look close enough, pretty much all surfaces of herbaceous plants have these little tiny hairs that stick out, and they'll perform different functions for different plants. You'll find, generally speaking, whereas the top of the plant is more wet, plant leaf is kind of waxy, the bottom of the plant where the stomata is, and this is a picture of a stomata, you'll find more trichomes there. And so the trichomes there are trying to slow air movement around the stomata in order to slow down that evapotranspiration, that loss of water. So it's like a little cool little adaptation that plants use in order to help conserve water. But also some plants you'll find are just like fuzzy, and that is like an anti-herbivore kind of tactic, you know, like nobody really likes chewing on something that's fuzzy, and animals don't. So yeah, so that's another kind of tactic that they can use trichomes in. Some plants will produce trichomes that are glandular. So they'll give off this like substance that could have some of this anti-herbivore property to it. Anybody who's touched a tomato plant will feel that and smell that glandular aspect of those trichomes. And I talked about trichomes last class, about in terms of on a tomato plant, their propensity to turn into like adventitious roots. Like they can start off as trichomes, but they can switch jobs and turn into a root system, which is kind of cool if they meet the right conditions. And then down here for trichomes and dermal tissue, you can see these really fine root hairs. Those are like the crazy amount of surface area that the dermal tissue, these root hairs, can form, and those are the main water absorbing and nutrient uptake kind of surface area for them. And then I guess to be fair, you could talk about the epidermis of woody plants is the bark, you know, so we would call that the epidermis for woody plants. It tends to be, if you think about cork, cork comes from the epidermis of a specific oak tree, and it has a lot of suberin. So whereas I was talking about the epidermis, the dermal tissue of a leaf, its job of keeping water in. Cork is obviously really good at keeping water in, but there's like this gaseous exchange. So those are examples of dermal tissue that you'll find in plants. And then I've talked about vascular tissue a little bit, but I haven't spent too much time of it. When we were talking about stems, we talked about how the stems of plants, they provide the housing for the vascular tissue, and how the vascular tissue is the plumbing of the plant. So if we break that plumbing down of the plant, the vascular tissue, the xylem is like a straw, which is always going in one direction. It's always taking that water with the nutrients dissolved in it up from the roots to the shoots. Whereas the phloem, we already talked about this, so I'm not going to spend too much time, is more of like this multi-directional kind of transportation system for materials. Depending on what time of year it is, then they'll be taking sap up in one direction or taking it down. And not just sap, there's other things that are called metabolites, other really important materials that they need to move around. And this is, I used the analogy of hydraulics in a car, because the phloem uses hydrostatic pressure in order to move these materials up and down. And I don't have time today to explain that. I'll be honest, I don't fully understand how the hydrostatic activity of the phloem works, but I am thankful for it because I like maple syrup. And then this one, it's not that exciting. This is the third group. It's called the ground tissue, but it is worth mentioning because it's going to come back. And there are 4 specialized cells that are in this group. There's the parenchyma, which has some metabolic function. They retain the ability to divide. They aren't fully specialized, but they do have the ability to become more specialized. And so, yeah, they can store energy. We were talking earlier about how stems have the ability to regenerate if they get injured, and they have an ability to store energy. So the parenchyma cells is where that's happening. And then there's the chlorenchyma cells, which forms bundles, like these flexible kind of bundles that allow, they give the plant strength, but they give them the flexibility to not like snap in a brittle manner if there's windy conditions, or maybe a kid comes and swings on the branch or something like that. And then there's the sclerenchyma cells, which are more, whereas these are the flexible aspect within the stems and the branches. The sclerenchyma are like the secondary walls that are more brittle, that give more of the structural strength to them. And then the sclerids we just find in the hard testa, the outside casings of seeds. So why did I tell you all that? Oh, there's one more. This one, I don't know, depending on who you read or what you read, the meristem tissue, it is the tissue that it is fully undifferentiated cells. They take place in these apical regions, whether it's in the root or whether it's right in the tip of the shoot. That is where this meristem tissue can be found. And so if you want to understand, like that's where the growth is happening. That's where you're going to find the meristem tissue. The analogy to humans is like stem cell tissues. There's all this, I mean, it's controversial research because they use fetuses as the source of the stem cell. But in animals, that stem cell is these cells that are brand new and they still haven't decided whether they're going to be, a specific organ in the human. And then for plants, it hasn't decided if it's going to be the ground tissue, the vascular tissue, or the dermal tissue yet. So this is really important for the purposes of explaining how to clone plants. If my slide will flick forward. No, I went backwards. Okay, so that was, now that we've talked about a whole bunch of things, the plant structures and functions and their life strategies, and now we've talked about those 4 different tissues that make up a plant as a whole, I think that gives us a good base to understand how to clone plants, how to take stem cuttings. And so because we've already talked about how the strategies of perennial woody plants, we know that in the late winter and the early spring, they're sending all those extra sugars up out to the apical tips. And that's what makes this time of year really favorable for cloning plants through taking stem cuttings. As you can see in this picture, this plant was sending so much sugar, it's emerged through that leaf bud. And then we've talked about meristematic tissue now. We know there's going to be a lot of growth right out at those apical tips. We know that the meristematic activity is pretty much at its highest point for the season. And so the way that we take stem cuttings is we basically just go up to a plant and right near the tip, about 4 inches off. I mean, you can do it a lot of different ways, but this is just one example here. And you can take about 4 inches to 10 centimeters, and you can just clip it with some pruning shears. And because we understand this awesome potential of meristematic tissue can turn into anything it wants, and we talked about how the parenchyma cells they are still a little bit undifferentiated. So there will be parenchyma cells down here. And they have that ability to divide. They have that meristematic potential. And so it might not make much sense to a novice gardener to see people taking a stick and sticking it in the soil and then having a brand new clone plant emerge from it. But once you start to understand all these processes and features of plants in unison, then it starts to make sense that you can actually culture a whole new root system out of what was the stem.
Can I have a question there?
Yeah, absolutely.
Yeah, I've tried this with lilacs, because back in Saskatchewan they used to give us stem cuttings from provincial tree farm, that they give us the cuttings and we put them in water for a few days and then plant them and they grow. Yep. And I've tried with lilacs around here for the last 3 years, and I can't get them to grow. But although am I cutting them the wrong way? I'm doing it around this time of year usually, not quite maybe this early, but about just before before they're getting green, definitely, like when they're still just buds. And I tried cutting them at different spots, and I don't know what I'm doing wrong, that they never have grown.
Okay, well, that's a good question. Yeah, I'll have to look into that, Jack. I took some stem cuttings of lilacs last year, and I had some success. Not a great amount of success, but yeah, I was able to culture a couple of them, and I don't know. But yeah, maybe there'll be something in the rest of this explanation that'll be helpful for you.
Okay.
And if not, then I'll do some more research before the next class and see if we can figure it out.
Okay, thanks.
Yeah, thank you. So I would say right now, if you were to take a cutting, it would be more like a hardwood cutting. And also, this is the book that I've learned how to take cuttings from. So it's called Creative Propagation, and there are cuttings for softwood cuttings, which would be more like in a month from now, right before they're about to bud out. And right now, if you were to take them, there's a technique more called hardwood cuttings, and so you take them at this point and you let the scars heal over. I've seen people put the hardwood cuttings into like a box full of dry sand, and you let those scars heal over, and then you do the rest of these steps. But like a softwood cutting a month or so from now, depending on the weather, obviously, because it changes every year, is kind of what I'm talking about right now. And the other reason why it's advantageous to do in the spring is this idea of the root to shoot ratio. I think I talked about how plants will generally keep the same amount of biomass above ground as they do below ground. And it makes sense because if they have stomata that are breathing out the water that they're pulling up from the root system, if they had a smaller root system than they had all the added up stomata in their leaves, then they would breathe out more water than they're able to take out. And so that lack of symmetry would cause them to wilt. So in the spring, if we take them at this stage, they don't have the leaves yet to breathe out. And so that gives that root system that we're trying to culture a little bit of a head start to get going before the leaves start to emerge. And the leaves can eventually overtake the root system, and we can cut them we can cut them in half in order to reduce that surface area. But just to have a equal root to shoot ratio to start is a little bit advantageous. This slide flipping thing. Yeah, there we go. So when you go looking for the appropriate stock, I mean, there's a lot of material that you could go out there and look. There's the Carigana, which I talked about last class as being in the legume family. So it's fixing nitrogen. It's a good bush shrub to clone. There's the lilac, which is just beautiful. There's the juniper bush, which the berries, even though they're not berries, they're succulent cones, are very high in vitamin C. There's spruce vine. There's all sorts of material out there that you could very easily take. But when you're taking it, you want to kind of avoid any deformations on plants or signs of disease, because those could be, when you're looking for appropriate material, you don't want to pick signs of weakness. You want to look for the most vigorous and healthy plants that you can. So something to take into consideration there. And then when you go out to take your cuttings, you would want to take some sanitized, make sure that they're clean so that you're not transmitting disease, any potential botanical disease between the plants, or you're not introducing a new one as soon as you take it. It's good to prepare your rooting medium beforehand. I've heard people talk about doing it in water, and I've only ever used like a really gritty soil, lots of sand, so that you can keep it wet, but the water is allowed to flow through really easily. There's lots of oxygen down around the roots. I use a little bit of peat moss as well, so it's draining, but it's also holding water as well. You need the gritty kind of sand in order to make sure there's pockets of oxygen, so it doesn't go anaerobic, but use a little bit of peat moss in order to keep some moisture as well. The cutting length that I like to go with is around, like I said, 4 inches to 10 centimeters. And that allows me to use like one of the tall domes to help get them going. I'll explain that in a second. And then when I go to take them, I'll bring a plastic bag so that if I'm gone for a while, the injured part doesn't dry out. It stays nice and succulent. It doesn't keeps its moisture. And so I'll bring them back, and this is the one part of the operation where I use a synthetic. So this is a synthetic rooting hormone, and it actually has a fungicide. You can buy one of these small bottles for around $5 at any garden center I've seen. And so it's a synthetic, it replicates a plant growth hormone. So you dip it in and you get a little bit on it, but you don't want too much. You want to shake off any extra. And so that's going to stimulate root growth. And again, because I wouldn't normally use a fungicide, this is the only time I ever do. But the fungicide keeps them from, it protects them from fungal infections. They're quite vulnerable at this stage. And yeah, so if you're a fancy grower, you might have a whole misting system set up. Once they start to leaf out, if that ratio of root to shoot ratio isn't right, then you can start to get the leaves will wilt and they'll die off. But in order to prevent that, a fancy grower will use a misting system so that the air around the leaves is kept so humid, it slows down that evapotranspiration to the point where that root to shoot ratio isn't problematic. If you're not a fancy grower and you're like me, then these clear domes are very useful because you can open them up and you can give everything a spray, get it all humid in there, and then you put the clear dome over top, and it keeps a very humid atmosphere around the leaves so that you don't have that wilting problem. But the clear domes can be problematic. It creates stagnant air all around your cuttings. So when I'm starting the cuttings, I'll take the dome off for maybe 15 minutes at a time and get some fresh air in there in order to get rid of those those potential fungal problems. Kind of the same way, Lou, as we were talking about with your microgreens the other day. Yeah, like Fungi is a really good thing down around the roots, but once we get it up around and stagnant air around our seedlings or our microgreens or our cuttings, that's when we can run into problems. So I like to introduce some fresh air periodically, once or twice a day, to avoid that. And the other thing about using a dome like that is you want to have them in a well-lit room. But not in direct sunlight, because you can have a runaway greenhouse. You can heat up really fast and just burn your cuttings to a crisp. So just in a well-lit room, but not in direct sunlight, is really good. And then it's very, very tempting to want to kind of test those cuttings and see if they've started to establish their root system. But the root system can be so kind of tender that if you tug on them and they've just started to catch, then you can destroy that progress and you can kill them. So the way that I've figured out, and I don't know how other people do it, because I've actually just been teaching myself how to do this over the last couple of years, but when I remove the dome, then after a couple of weeks, and I think they're starting to take, when I remove the dome, I'll remove it for a longer period of time, and I'll be close at this point, and I'll watch them. If they start to wilt, then I'll know that the root system isn't quite established yet. So I'll put the dome back on, and then I'll wait a couple days, and I'll kind of do that. And once they stop to show those signs of wilting, that's when I know that they're good to go. And there will be, you know, you're not going to culture the roots on every single cutting, but this is, you know, so there is going to be a level of failure. I've been told I shouldn't use that word, that's too harsh, but like, it's not going to be a perfect method. But honestly, the cost of a bottle of this root stimulant is $5. So like the risk reward ratio, even if all your cuttings don't make it, the risk reward, you can, in a small space, you have the potential to start so many of these plants. It's fun to watch, and it's affordable, and it's a great lesson. And then I guess I should have mentioned this one, rosemary. I think a lot of us want rosemary in our gardens because it's such a wonderful culinary herb, but it's really, really hard to start from seed. So this, I don't recommend starting rosemary from seed. It takes really well from these stem cuttings. And so, yeah, that's my spiel, everybody.
Great, thanks, Christian. I feel like all these foundational building blocks are kind of starting to come together. I can feel it.
Yeah.
Excited for the next class too. Katerina had a question in the chat. What's the best way to know when to plant? When the last frost is? Is there something in nature that would be a good indicator?
Yep. Good question. I actually, the best advice I ever heard on that, and obviously it comes with the caveat that some plants are frost sensitive. So for the frost sensitive plants like your zucchini, or even your basil, or what's another one? There's a whole bunch. I have a list. I'll be putting that list up again. For those, you would want to wait till around the 3rd week of May. That's usually our frost-free date, but you're looking at the long-term forecast in order to make sure about that. And then on the other end of the spectrum, there are those really hardy, cold-tolerant crops like your spinach and your peas and your radishes and mustard greens. And again, I'll share the full list of those. But the best advice I ever heard about like that, what's that thing in nature that you look for to know that it's safe to plant those really cold hardy ones, is a farmer from Highwood Crossing just south of Calgary. Tony, he told me that when you see the weeds sprouting, that's when you know. So like once you start to see some weeds around town or in your garden, then you know that those really cold hardy crops can be planted. But I would also say, like, yeah, it's great to get them in, and you can get earlier productivity. But I have had like these times where I look at how slow they're growing in the really cold times, and because I was growing on like a quarter acre, the work that I had to put in to keep them going and them growing a lot slower, it was like there was a ratio of work to production that I was like, I could just wait a couple more weeks and they'll probably be ready to harvest at the same point. So I have done that in the past where I've seen the weeds sprouting and I'm like, all right, I'm getting the radishes in, I'm getting the peas. And it works, but they're not thriving to the same extent where you can kind of save yourself energy and your time and just wait a couple more weeks after that and put them in. Yeah. But yeah, again, I want to preface that with like the context, because if you have a garden on your balcony or like a really small south facing garden, then that work to reward ratio is different. It's just like a tiny little spot, and the amount of work that I have to do to cover everything and take the covers off to water them is not the same as if somebody has a small garden and you want to get production going early. It's a lot more work efficient in that sense. So take from that what you will.
Good tips. Any other questions, comments for Christian? I don't see anything else coming in. What's our next class on Monday all about, Christian?
Our next class on Monday is about my favorite thing in the world. It's about soil. So we've talked a lot about plants. We could talk more about, but soil is so important. Those interactions with the root system. When I started gardening, I didn't know very much about plants, but I knew enough about soil. And if you have really good soil conditions, it makes growing plants really easy. So.
Awesome. Question just came in. Do you do composting teas?
I haven't done teas. I've done extracts. So a compost tea is where you put compost into water and you actively aerate it, and that shakes out beneficial microbiology. But a tea is where you actually feed that biology. So if you wanted to make a tea that was full of bacterial life, then you would put like molasses or something sweet into it and brew it up, and that would actually breed more bacteria. And then on the flip side, if you wanted to make a tea for some fungal partners, then you would actively aerate it with like some fish hydrosylate or some emulsions, and that would feed the fungi. So I've never made teas, but at the golf course with the vermicomposting program that we set up there, we have done extracts where it's just strictly putting in the high quality compost. And we know that high quality compost has a lot of these really beneficial microbiology that are going to help the plant, and it just shakes all that out. And then you can spread that basically probiotic yogurt through your garden. You can make more of it than if you were just to use like the compost you started with. So
Great, thanks. Don't
Who asked that question about teas?
What's that?
Who asked that question about teas?
I think it was a Katerina. Yeah.
Okay.
Mhm.
I'll include some resources about making teas and extracts in next class.
Sounds great. I don't see any more questions, but I see a lot of thanks in the chat, and everyone is just in awe of the amount of knowledge you have. So thanks again, Christian, and thanks everybody for tuning in again, and we'll see you next Monday.
Yeah, thanks everybody. Thanks, Lou.
Thank you, Christian. Bye.
Bye-bye.